Good morning. Happy midterm week Monday. I'm sure we're all excited about that. Only stuff that's going to be on the midterm in this class is stuff that we cover today, and of course everything we've covered up to today. Material on Wednesday will not be new. As far as the midterm is concerned, get here on time. As long as you get here on time, you can have as long as you need to finish the midterm. But as soon as the first person leaves, when they're done with the midterm, then no one's going to be able to start it. So be here on time on Friday. That'll start at 9. Any questions about the midterm at this point? Yeah?
Are you expecting it to take more than the time?
So the question is, do I expect it to take more time than that? No, I expect it to take about half an hour. Usually the people who finish first take about half an hour. So it should be twice as long as you need. But some people want longer. I have plotted how long people take and how well they do. It's a flat line. So working on it longer, you're just as likely to change the questions to the incorrect answer as you are to the correct one, at least on average. Other questions? Yes? Yes, you do need scantrons. Other questions? No. Okay. The other thing? Yes, sure.
Is there any way to make the room more
Today?
Right, yeah.
Can somebody hit the lights over there, please? Someone near the light switches? I haven't figured them out in this room yet, so I'll let somebody else do it, and it could be their fault. The stuff today should be fine in terms of lighting. One other thing, yeah, that's good. Yeah, more, I think. Is that too much? I think that's going to be okay. It'll be better for our sign language interpreters here, too, I think. Good. Clicker questions. No, I've not posted them yet. I'm going to try and get to that today, or your answers, I should say, for the clicker questions. And that being, of course, a wonderful lead-in to our clicker questions for today. So, clicker question 1, which RNA binds directly to mRNA? Say rRNA, I should say. 16S, 18S, 23S, 5S, 5.8S. So during these questions, sometimes I wish I could walk around the room and listen to you and help out with some of these things. I think that'd be great, but there's one of me and 106 of you who voted. So that makes it a little bit difficult. So what do you think? Almost all of you thought 16S, which is correct, because that's the small subunit RNA in bacterial ribosomes. Binds to the ribosome binding site. And so also known as the Shine-Dalgarno sequence in the messenger RNA. So that's what that is. The 18S is the small subunit ribosomal RNA, the main one in eukaryotic cells, but that doesn't associate directly with the messenger RNA. It's all of the other components in the pre-initiation complex that do that. 23S is one of the large subunits. These smaller ones here, 5S and 5.8, don't directly interact with the messenger RNA at all. So let's select the correct answer. Good. And then move on to our next, which is the initiator tRNA binds to the blank site or sites of the ribosome, A, P, E, A and B, or B and C. And when I say A and B, I mean the A site and the P site, and B and C, the P site and the E site. Okay, what do people think? Now there are 110 of you. A few more showed up. Almost everyone seems to think that it binds to the P site, which is correct. It never, ever binds to the A site. Why is that? Okay, the A site always has to be open for the next one to come in, which was a comment from the audience here. But what else? It's really critical about this. The initiator tRNA is not the one you use to put methionine in the middle of your protein. So it's always going to have to go into the A site. What happens when the ribosome translocates after you've put it in the P site, and then you put something in the A site and you make a peptide bond? Where does that initiator tRNA go? The E site. So that would be the P site and the E site, which would be B and C. So obviously I did not do a very good job of explaining this. There's only one of you got it or guessed it, as the case may be. Yes, technically it does go to the E site for elongation. It doesn't say anything about initiation here. It's the initiator tRNA. So our next clicker question, assuming we can actually get it to move here, is in translational elongation, GTP hydrolysis is used for ribosome translocation, correct tRNA codon interaction checking, peptide bond formation, A and B, or A, B, and C. Okay, now we've got a huge difference of opinion all over the place. So, at least most people thought it's peptide bond formation, correct tRNA codon interaction checking, and ribosome translocation. Guess the next one thought ribosome translocation. Ribosome translocation is correct. What protein is involved in doing that? In bacteria. What protein? Alphabet soup. So it's an EF for elongation factor. Which one? G, correct. So EFG is important for ribosome translocation. And it's a GTP, ACE, will hydrolyze GTP. What elongation factor is important for tRNA codon interaction checking? EFTU, correct. Does that hydrolyze GTP? Does it? You've already voted. No. Yes, it does. So it hydrolyzes GTP, and that's a conformational change. That's how you know you've got the right tRNA, which is there at the A site. Because you've got a bunch of wrong ones. There are at least 20. Most organisms more than 20 different tRNAs that could be binding in that A site. So they're going to be basically selecting through. The ribosome is just sitting there waiting for the right tRNA to come in. And the way that it knows it's the right tRNA is because EFTU, or eukaryotic elongation factor 2, will recognize that that's the correct base pairing interaction with the codon, and then have GTP hydrolysis. So I guess I did not do a good job of explaining that. Peptide bond formation. What does peptide bond formation? What's the enzyme that does that? The ribosome, exactly. So ribosome hydrolyzing GTP. No, the ribosome is not hydrolyzing GTP. So what does that leave us with? Eventually we'll get there. D. So it's ribosome translocation, correct tRNA codon interaction, so EFG and EFTU in bacteria, eukaryotic elongation factor 1 and eukaryotic elongation factor 2 in eukaryotic systems. But peptide bond formation has nothing to do with that. Where does the energy come from that's used for peptide bond formation? Amino acyl tRNA synthetases, exactly. And what does that use for energy? It uses ATP, exactly. So with that, we'll move on. I think we've mostly reviewed translation in as much as we need to review translation, I think. Initiation for bacteria, I guess the big difference, and the things we didn't talk about in these clicker questions, has to do with the difference in eukaryotic versus bacterial messenger RNAs, linear polycistronic versus basically circular, usually monocistronic transcripts, and then how you identify your start position, either 16S or all of the eukaryotic initiation factors, bind to the cap, translocating along the RNA. Some people have asked me, which of that humongous long menu of initiation factors do I expect you to remember? I expect you to remember the ones that we talked about in class. So these would be the EIF4F complex, and then those individual ones, particularly the ones that we just talked about. There are a whole bunch of other ones which are really fascinating, love to talk about, but I can't expect you to remember them. Elongation and termination. Are there questions about that? We've got quite a bit of replication to go through today. So, okay, if there aren't questions, let's move on. In terms of replication, we're going to talk about how you go about doing replication in terms of substrates and enzymes. Hopefully all of you know that DNA replication is semi-conservative and semi-discontinuous. We'll look at that sort of where people have more issues in terms of the primers, origins, initiation, and licensing. So what's the template for DNA replication? It's, not surprisingly, it's our friend, the double helix, which we had, and we can actually get this to move. That would be nice. Here we go. Predicted by Watson and Crick. So right at the end of their paper, which all of you have read, yes, good. They have the quote about, "is not escaped our attention," that this has fundamental implications for replication of the DNA. I think one of the best understatements in molecular biology. And basically what they say is, well, we think that these two strands are going to come apart and each of them is going to get replicated. And so this was their immediate prediction. And they also had that both of these strands would be copied. So they immediately predicted that you were going to have replication by pulling the two strands apart and then replicating them. They actually thought that they were going to be replicated at the same time in the same direction. So they were wrong in that, one of the few things that Watson and Crick were wrong about. But a template, again, the standard template that we've already talked about. Not quite sure why this is not advancing, but it does eventually. Let's see. Yeah, we'll just do this here. So there are really multiple different replication models, which came up basically after Watson and Crick had made their prediction, but also these were kind of straw men that were set up in terms of being able to test them with experiments. And the idea here is, okay, semi-conservative replication, which you have your two strands of so-called parental DNA, you pull the two apart, you copy each of those strands independently. So this red one is new DNA. Another distinct possibility in terms of how this could have been working is that two strands don't come apart. You just make a copy. And we already talked about major groove. You've got all the information that you need in that major groove. So in theory, you should be able to go from a double-stranded copy to a double-stranded copy without pulling the two apart, etc. And this is actually really quite an attractive model, and a number of people liked it at the time. Another possibility is you just mix and match. You take little pieces of the DNA, pieces of each strand, and copy each of those. And it turns out that this is exactly what happens in DNA repair, is you do this dispersive replication. You take little pieces and copy each of them. And so this was a really quite reasonable kind of way to think about how replication could have been taking place. So, of course, hopefully all of you know the so-called most beautiful experiment in biology by, in fact, a teacher of mine. He wasn't, actually, I was TAing for his class, a fellow by the name of Gunter Stent at UC Berkeley, who tried to do this experiment and failed, which also gives you an idea how recent a lot of this stuff in molecular biology is. He tried to do exactly the same experiment, only used P32 instead of N15, and I'll let you think about why it didn't work. It's a really great thought question to think about. So, in the Meselson-Stahl experiment, basically they were trying to separate between these three different models. You had semi-conservative replication, conservative replication, and dispersive replication. And how to go about doing that? Well, basically, this was one of those wonderful collaborative experiments that worked. Matt Meselson was a real expert in the separation technology using ultracentrifugation, and Frank Stahl, who just recently retired from U of O, just down the road, was really good at doing bacterial growth experiments. And so between the two of them, they were able to come up with this, again, most beautiful experiment in biology. Basically what they did is they grew their favorite organism, in this case E. coli, in heavy nitrogen. This is the N15 label. And then moved those cells, and this was the key part I think that Frank Stahl did, was moving those cells in such a way that they kept growing completely normally into light nitrogen, so N14, and then just waited and saw how much of each of their DNAs was replicated over time. The data are shown over here on the right-hand side. When you start out, you have heavy, and this was a cesium chloride equilibrium density gradient. You have a band over here, and then as time goes by, you start to get this middle band right here of intermediate density. And there are two really important things about this. The first one is that you've completely lost this heavy band. And so that means that your conservative replication model's out the window. On the other hand, dispersive would give you exactly the same thing here because it's half and half. Makes no difference. So they have to continue the experiment until they get to their second generation, which is right here, and then you start to see a separate population which is just light molecules. And what that means is, again, down here at the bottom, is you've got these semi-conservative, you've still got this, and actually, oh, this is interesting, the new strands here. So the new strands are blue, the old strands are in red. You're always going to have one strand in your duplex, or half of it which is old and half which is new. But as you keep going, of course, that new half is going to get copied into new, etc. And so you're going to end up with more and more of this light band here. And so the only interpretation of this experiment was that DNA had to be replicating in a semi-conservative mechanism. So hanging on to one strand and making a whole complete new one. What does that? So what does that? It's the DNA polymerases. Again, not surprisingly, these are the enzymes which will take and copy each of those strands. Now, one of the big surprises that people had, which was actually pretty soon after a lot of this modeling was done, this is 1957, that Arthur Kornberg's lab discovered this DNA polymerase. What they found was that these DNA polymerases always attached a nucleotide triphosphate to a 3' OH, and always are going to polymerize in a 5' to 3' direction. Now, this is a problem, of course, because the double strands of your DNA are opposite to each other. So 5' to 3' is really nice if you're going to be replicating one strand, but it's a real pain if you're trying to do the other one. And apparently Arthur Kornberg used to go into his lab every day and say, "Where's our 3' to 5' polymerase? We need this polymerase." And they never found it. And there are important reasons for that, too. A couple of other things. Of course, it needs a template. And that's not terribly surprising. But the other big surprise about these DNA polymerases is that they always had to have 3' OHs to extend from. And this is really common for all of the DNA polymerases. They have to have OHs. And this should immediately be telling you something which is, well, that's kind of odd, because if you have to have a 3' OH, this kind of means that where's the egg that this chicken has come from? So it always leaves you with this, this problem. So let's look at that mechanism, however, the mechanism that Arthur Kornberg, first he found DNA polymerase 1, then DNA polymerase 2, and DNA polymerase 3, in that order. All that they do is they take deoxyribonucleotide triphosphates, like this one right here, which base pair to whatever the appropriate nucleotide is on the template strand, and then catalyze this hydrolysis right here of the alpha to beta bond of your nucleotide triphosphate, causing the release of pyrophosphate. And a couple of people asked me about what was pyrophosphate last time. Pyrophosphate is just these two phosphate residues that are hooked up next to each other. And so that release of pyrophosphate, which happens. And then again, you're always adding to the 3' end because it's this phosphate group from your nucleotide triphosphate that gets attached to that 3' end. So this is, again, it's great and wonderful, but the big problem is that, well, how do we get that other strand to go? And hopefully, this is all review for you, is that, fine, you've got your DNA polymerase, which can go from 5' to 3', but for your daughter strand, you have to make it in little pieces. And this was shown by Reiji Okazaki, who ended up using huge amounts of radioactivity and dying relatively young from leukemia. Was also in Hiroshima when it was bombed. Whether there's any connection between those things, we don't know. But at least the Okazaki fragments are still named in his honor. So he found that there were these short pieces of DNA that were getting made at the same time as that long piece of DNA. And he actually didn't call them leading and lagging strands. That came along considerably later, that people came up with this. So what's actually doing these jobs? It's not just the DNA polymerase. You also have a number of other enzymes that are important here. Okay, in bacteria, it's mostly the DNA polymerase 3 which is doing the job, not DNA polymerase 1 that was found originally by the Kornberg lab. We'll talk about why that is in just a second. You also have a DNA primase, helicase. In the case of E. coli, this is the DNA-B protein, single-stranded binding protein, beta clamps, and clamp loaders. So this is always present at the replication fork. We've got a leading and a lagging strand coming together next to a double-stranded template. You have to have all of these enzymes. These are just the names in bacteria. The equivalents in eukaryotic cells are DNA polymerase delta, also DNA polymerase epsilon. Gives you an idea how many of these different polymerases have been found. There's also a DNA primase together with DNA polymerase alpha, the so-called MCM complex. Each of these guys do the same thing here, helicase. RPA is replication protein A. It's a single-stranded binding protein. PCNA. PCNA is really confusing. Why do you call it PCNA? Because it's proliferating cell nuclear antigen. Comes from cancer research. When if you look at cells that are growing really rapidly, like cancerous cells, they have a huge amount of this protein. And we'll see why that is in just a second. And then replication factor C, which serves the same job as the clamp loader process. What do replication forks actually look like? Here's an example from a viral sequence. This is the SV40 DNA. Take virology if you want to learn a lot more about SV40. But basically, it's a circular DNA which has one origin of replication on it. Wonderful model system for understanding eukaryotic replication. And a lot of times in textbooks, you'll see eukaryotic replication forks. They're really SV40 replication forks. So here you have the origin of replication. It's now in a linear piece of DNA because this has just been chopped at one particular position to give you a linear DNA. And you see this, what's called a replication bubble right here in the middle, which then just gets bigger over time. And that means you have two replication forks, one at this end of the bubble and one at the other end of the bubble. And that's nicely shown here in this slide from your textbook. Helicases, the really critical part about getting replication to start is pulling those two strands apart. If you can't pull the two strands apart, you can't replicate because the DNA polymerase has to have those free nucleotides on the templates to work with. And so if you think about DNA replication, the critical aspect of that is pulling the two strands apart. Once you pull the two strands apart, you're going to get replication. But you have to get those two strands apart. To do that, again, you need these DNA helicases. I don't like this slide that much, again, from this textbook, because these should actually just be around one strand of DNA, not around both strands of DNA. But they pull apart the two strands. And then once you've had these two strands pulled apart, you're going to have leading strand primers which are synthesized. And again, these are two opposite strands, but you've got two replication forks. So you'll have two leading strands that are being made here. And then you're going to have lagging strands that fill in at the ends of each of these. So right at the origin, you're going to have two leading strands going away from each other, and then the lagging strands which are right next to those. So that's great and wonderful, but I mentioned before, DNA polymerases are really wimpy. They have to have a primer or a 3'OH to actually work. Well, where do those primers come from? Back to our friend RNA. So all of the primers, and this is not just for the lagging strand, but also for the leading strand, that first primer that gets put down right at the replication origin, that's from RNA. So it's an RNA primer. And RNA polymerases, like we talked about when we talked about transcription, those can start in the absence of an OH. They can start from scratch. So RNA polymerases can do that. So we have an RNA primer. That RNA primer provides a 3'OH, which then the DNA polymerase can come down and extend. And so here we've got our new DNA in red. Here's our leading strand. There should be an RNA primer over here in green. The RNA primer down here on the lagging strand. That's going to get extended, and then you have to put another primer here. That will get extended. Put another primer here. That will get extended, etc. What does the job of putting on these primers? This is the aptly named DNA primase. Confusing, however, a little bit sometimes because DNA primase is actually an RNA polymerase. So it's something to bear in mind again. So your DNA primase is an RNA polymerase because it starts with no primer and puts down a nucleotide triphosphate, extends that till you get a 3' OH, which is now base paired with your template. That now is a wonderful substrate for the DNA polymerase to extend from. A couple of problems, however, with these RNA polymerases, which are the DNA primases. First, of course, they're RNA polymerases, and we know that our genetic material is double-stranded DNA. So somebody got to get rid of that RNA. And the other problem that you have with DNA primases is they're incredibly low fidelity. And what I mean by low fidelity is they often put in the wrong nucleotide. And if they're putting in the wrong nucleotide, you don't want to have that piece left in your genetic material. And so you need to get rid of these guys somehow. And this is a process which uses, not surprisingly, nucleases, but also that DNA polymerase I, which Arthur Kornberg's lab first found. And then finally, you have a DNA ligase. What happens here? Well, the first thing that happens, you have RNase H. Now, RNase H, the H stands for hybrid, hybrid between DNA and RNA. So, RNase H chews up RNA, which is in an RNA-DNA hybrid. Really quite straightforward. Breaks it up at the phosphodiester backbone. These guys fall off. Then you have DNA polymerase I, which will basically fill in these gaps. And so it should be down here. The DNA polymerase I right here is going to be binding to this piece of DNA here and extending this primer. One thing about DNA polymerase I that we'll talk about more when we talk about repair after the midterm, so you can turn off your brains for the next 2 seconds because, hey, it's after the midterm, I don't need to know it now, is this DNA polymerase I also has an exonuclease activity where it chews up pieces of DNA, and particularly what it does is it has an exonuclease activity which is going from 3' to 5'. And so it chews up the template which is in front of it. And that turns out, of course, to be really nice if you chew up the template which is right in front of you, which has to be it's RNA, and often is the wrong RNA because the fidelity of that DNA primase is relatively low. Last thing, however, you need to do is hook these two ends together. You've got a 5' phosphate here and a 3' OH here. That's the ligation, the DNA ligase, which does this step. This is a classic enzyme. What does it do? It uses ATP and the energy of ATP basically to activate the phosphate, this 5' phosphate that you need to modify. Or they're going to hook up to the 3' OH. Goes through a nucleotide protein covalent intermediate, just like we've talked about for our protease enzymes, many of the other enzymes that we look at. There's going to be a covalent intermediate that takes place. This then, covalent intermediate, activates the 5' phosphate here. And then once this is an activated phosphate group here, this adenosine monophosphate is a wonderful leaving group. This OH attacks this diphosphate, and then you lose AMP, you ligate your DNA back together. So you need, of course, lots of these enzymes, the DNA ligase, the RNase H, and the DNA polymerase I, because every time you put down a primer on the lagging strand, you're going to need to get rid of it. And that's the DNA polymerase I, the RNA ligase, the DNA ligase, and RNase H. So that's why you've got much more of the DNA polymerase I inside your cell, because you've got to be working on your lagging strand a lot more than the DNA polymerase III, which is the one which is making your leading strand. It turns out also making some of the lagging strand as well. And we'll look at that a little bit more a little bit later on. So, yeah, sure. RNase H.
And all one both chew up the phosphodiester bond of the primers?
Yeah, so it seems that they're both are doing that job, and so they seem to be somewhat redundant relative to each other. And the idea there is that it's such an important thing that you've got to get rid of that primer that you have two ways of doing it. Yeah. And molecular biology is cool. I agree. So replication, just like transcription, just like translation, 3 different steps that you have. Fortunately, they're a lot less complicated, confusing, extra proteins that are involved in that whole process. Initiation, again, this is critical. You need to know where to start, and you need to get those helicases on the DNA, because that's that critical first step that has to take place. Once you've done that, elongation is actually really pretty straightforward, but we talked about that semi-discontinuous nature, again, because all DNA polymerases can just add to 3' OHs. They can't go any more than that. So you have to have semi-discontinuous replication, and then you've got to stop somewhere. Well, how do you stop? That's when these 2 replication forks actually bang into each other. And if you think about a circular genome, that's going to be at the end of where you started from. In fact, the opposite end of your circular genome from where you start. Where you get multiple replication forks running into each other, and termination in eukaryotic transcription is actually extremely poorly understood and a really active area of research right now. So let's look at a cartoon of how initiation works. We also know the most about initiation. Basically, you have a double-stranded piece of DNA, and somehow you need to tell the replication machinery, which of course is mostly those helicases, to get to the right place. So normally what you have is you have DNA binding by an initiator protein. This is a very generalized cartoon here. And once you have your initiator protein being bound, that leads to the 2 strands coming apart. But 2 strands coming apart is relatively easy. They're going to go back together really quickly. The really critical part here is that you now have to get other proteins here, and most important of those is that DNA helicase, which here is appropriately shown as just being around 1 strand of the DNA. And again, we'll look at that helicase in much more detail as we move along here. So what do these origins of replication look like? For most bacterial chromosomes, which are circular chromosomes. Oh, yeah, question.
So the DNA helicase doesn't necessarily do the unwinding, it's just holding the unwound DNA apart?
Okay, so we'll talk much more about the DNA helicases in just a second. But basically, those 2 strands coming apart in the first place, you need some helicase activity to do that, but that's just the start. And to actually move away from your origin of replication, that's where you really need the DNA helicases for. Well, that's kind of getting ahead of ourselves, but yeah, that's where you're going to need it. So here, 2 strands come apart, and then you really need your helicases to pull these 2 strands apart, as we're making now 2 replication forks, each of which go around the genome. Now, that's great and fine and wonderful if you have a circular genome. But as hopefully all of us know, our genomes, and the true for all eukaryotes that I know of, although there could be some exceptions, have linear genomes. And so how do you deal with that? Well, it turns out that our genomes have multiple origins of replication, each of which, however, proceed in the same kind of bidirectional 2 replication forks starting at a particular origin and moving away from each other. Let's first talk a little bit more about the E. coli situation. But again, these are things that you need for everything. You always need an initiator. You always need a helicase. You always need a helicase loader. And if you notice, it's a ring around the DNA. Well, how do you get a ring around a single strand of DNA if you start out as being double-stranded? You need some very specific proteins to do that. And these are the so-called helicase loaders. And then once you've gotten those two strands apart, the DNA polymerase can't do anything until it's at a primer. So you have to have a primer. In the case of E. coli, your initiator is DNA-A, helicase is DNA-B, helicase loader is DNA-C, and DNA-G is the primase. And again, I apologize for the alphabet soup, but this is really standard in the literature. So what do these guys interact with? They interact with the origin of replication. In E. coli, it's called ORI-C, or the chromosomal origin of replication. There are also ORI-Ps for plasmids. You talk about viruses, there'll be ORI-V. So ORI-C for E. coli, there's just one of them in the circular genome. Anybody remember how big the genome is of E. coli? About 5 million base pairs. One place to start. E. coli can replicate really fast. So I'm making more cells, half an hour, 40 minutes to half an hour, which means you've got to replicate this whole genome from one start position in about 40 minutes. Those DNA polymerases are cranking to get through all of these things. So origin of replication, again, bidirectional replication forks. Once you get to the end, you have termination. And these two guys have a problem because they're connected to each other. Two double-stranded circular DNAs. What do you need to get those two apart? Topoisomerase. What kind of topoisomerase? Topoisomerase 2 to pull them apart. But that's not the only thing. You're pulling these two strands apart. You're untwisting them as you're going along. What does that mean you also need? Topoisomerases as well, because you're pulling these two strands apart. You're untwisting them. So you also need to deal with all of these turns, and this particular calculation is assuming that you've got a 4 million base pair genome and 10 nucleotides per turn, so 400,000 turns in about 40 minutes that it takes to replicate. So lots of topoisomerase activity. This is a reason that, in fact, I talked about the antibiotics last time. Antibiotics work on translation. The other big set of antibiotics, they work on topoisomerases to block that particular activity, because you need them. If you don't want to replicate, you have to have a bunch of topoisomerase activity. So this is what happens. What does that origin itself actually look like? This is the ORI-C in E. coli. Has lots and lots of binding sites. I don't expect you to remember all of these names. Tau1, Tau2, R5, I1, etc. Important here is you've got initiator binding sites, and the initiator in E. coli is DNA-A. So that binds to all of these DNA-A sites. And right next to it is this region that's easy to unwind, also called the DUE in this particular textbook, but it's not a pretty... So DUE just means it's easy to unwind these. Not surprisingly, these are very AT-rich, because ATs have only 2 hydrogen bonds between the two strands, whereas GCs have 3. So this AT-rich region right next to where you have binding sites for this initiator protein. So one other thing I wanted to mention here, and that's other binding sites for things which are not the initiator protein. It's called here IHF and FIS. Basically what these are proteins which bind to and bend the DNA into a very specific structure. And so the structure of DNA is really important in terms of how DNA-A is interacting here and then stimulating the unwinding which takes place right here. So this is all well and good. It's great. You've bound your DNA-A there. You unwind the two strands of DNA. The helicase jumps on there and you take off and you run around the genome, do your replication thing. But one problem happens if, well, you've started there, DNA is still around. How does the cell know not to start there again and make more from that replication fork and make more from that replication fork and make more from that replication fork? That could be a real problem if you're trying to divide and get one chromosome per cell after you've done that. So this brings up the concept of what's called licensing. And all that licensing means is, hey, I've used this origin. Don't use it again. And the way that this works in E. coli is DNA, once it's been replicated, is methylated. We'll talk about DNA methylation a lot more later on in the course. But as far as this is concerned, DNA is methylated. Both strands are methylated. Those two strands come apart. You make new DNA. And so in a manner actually kind of equivalent to what Meselson and Stahl were doing, now we've got double-stranded DNA, some of which is old and some of which is new. The way that you can tell you've got half old and half new is that you have one strand which is methylated, the other strand is not methylated. Because it takes longer for DNA methylation to take place than replication to take place. So if you've got hemimethylated DNA at your origin of replication, you've got all your DNA A sites, your DUE, etc. If that's hemimethylated, one strand is methylated, the other strand isn't, you know not to use that again for an origin of replication. Until later on, when you've methylated both those strands, you say, okay, now I can go back and use these for replication again. So this is how E. coli knows, and right at the end we'll talk about how eukaryotes know about this. Turns out it has nothing to do with DNA methylation whatsoever. So once you've taken these two strands apart, what do you do with them? Well, what you need to do is put the helicase on them. So here we have DNA A, which is up at the top here, is sort of this lollipop-shaped structure, these two different pieces here. These guys bind to their DNA A binding sites and then cause the two strands to come apart right here. And this is actually not the normal DNA helicase that we talked about before. This is that DNA A actually has a small amount of helicase activity. So it pulls the two strands apart, and then you have to get the helicase, DNA-B, bound onto one of those strands. Now again, it's a circular molecule. We'll see pictures of it in just a second. Getting that on is tough. And again, it's okay, it makes sense, because you don't want to replicate all the time. So you need to have the clamp loader complex, which is what's shown here. You have N-terminus and C-terminus is not that critical. This helicase loader then has to come on. You load the helicase. Loading is just putting it on, getting that circular structure around the DNA. Once that's happened, now you get the primase that associates with this helicase, and now primase makes your primers, DNA polymerase comes in and extends from there. Again, the real critical part of this puzzle is the DNA-B protein or the helicase. This guy burns huge amounts of ATP, because it's got to be pulling apart those two strands. Goes at about 1,000 nucleotides a second, so again, it's a really rapid molecule here, and generates lots and lots of single-stranded DNA. But again, it's that loading process of this replicative helicase, the one that moves really long distances. That's what's key in terms of getting your initiation to take place. So, goes 1,000 nucleotides a second. It's wrapped around that strand of DNA, so it's not going to be coming off. It actually, once it's on, does a great job. On the other hand, the DNA polymerases themselves have another problem, in addition to the fact that they have to have primers and templates to work on. And that is that all DNA polymerases by themselves are non-processive. And what I mean by non-processive, talk about this a little bit when we talk about RNA polymerases. RNA polymerases are incredibly processive. DNA polymerases are not processive. They fall off DNA all the time. So what normally is going to happen with most DNA polymerases is you'll add a couple of nucleotides, and that DNA polymerase will fall off. And then another DNA polymerase will come on and add another nucleotide, and it'll fall off again. That's not terribly efficient, particularly if you want to replicate like the E. coli genome, 5 million base pairs in the amount of time it takes to replicate a whole cell. So you have to have another way of dealing with this. Now, the way that this happens, again, it's not terribly surprising. It's what's called a processivity factor, and that processivity factor, just like the DNA helicases, loops around the DNA. It's another circular structure which goes around now the double helix of DNA. And basically, all that it does here, this so-called sliding clamp, is bind to those double strands of DNA and hold on to the polymerase. So when the polymerase tries to dissociate, it stays associated because it's bound through protein-protein interactions to this sliding clamp that's stuck on the DNA because it's wrapped around it. These structures, again, are literally rings around the DNA. Their structures are almost identical in 3 dimensions between bacteria. This is a virus. This is the eukaryotic proliferating cell nuclear antigen. All of them have very similar structures. Turns out that they're made up quite differently in terms of the individual polypeptides, but same structure, same function. What is that function? Again, when that DNA polymerase tries to dissociate from the DNA, it's just held on through these protein-protein interactions with the sliding clamp. Once, however, you get to one of these primers at the end, that causes dissociation of the polymerase. It gets stuck. It can't go any further here, unless, of course, you're a DNA polymerase 1, because DNA polymerase 1s have these 5' to 3' exonuclease activity, so they can chew up this end piece here. Well, that's great and wonderful, but what if your DNA polymerase 1 chews up all of your strand? If your DNA polymerase 1 were processive, what would happen? You'll eat up all of your just made new DNA. So that's a real problem. Well, it turns out DNA polymerase 1 doesn't associate with pCNA. It falls off. It's non-processive. And it kind of has to be non-processive, because if it wasn't non-processive, it would chew up all the DNA. So that's what's going on in that case. The other thing that's formed by these helicases and what you have at replication forks are single-stranded DNA. Single-stranded DNA by itself is incredibly unstable, so you have to do something with that single-stranded DNA so that it either doesn't come back and fold up on itself, like RNA does all the time, or get degraded. These are single-stranded binding proteins. The single-stranded binding protein in E. coli is very conveniently called SSB, and very confusingly in eukaryotes called RPA. But these guys have basically identical structures to each other, and they play exactly the same role, blocking secondary structure formation in your DNA and making sure that it doesn't get degraded. Single-stranded DNA binding proteins bind in what's called a highly cooperative fashion. Basically, once one of these proteins is bound, you get a whole bunch more of these proteins bound, one leading to many very rapidly. We already saw cooperativity when we looked at the 2 DNA strands coming apart. You just start a little bit with the DNA strands coming apart. They come apart, the whole thing, very rapidly. So it's really the first binding event is the really critical one. That determines your rate. Once that's been bound, then all the rest of them bound actually really, really rapidly. So termination. So we talked about initiation, this elongation. You got your helicases, you got your sliding clamps. Termination. In E. coli, it's really easy because it's when the 2 replication forks come together. But we've got multiple replication forks that we have in eukaryotes. So last 10 minutes or so, let's talk about some of those differences in eukaryotic replication relative to what's going on in bacterial replication. First, again, linear chromosomes. Okay, that makes it clearly different, certainly in terms of termination. Multiple origins of replication, and we'll see why that is in just a second. Multiple DNA polymerases, so not just those DNA polymerase 3, which is the one which is doing all that elongation process, and DNA polymerase 1, which chews up the other strand. You got about 6 or 7 DNA polymerases in eukaryotes. And then, of course, you've got to deal with ends somehow. Once you've got to the end of your linear piece of DNA, you don't have primers, you end up with telomeres. It's a mess. We'll talk about that much more later on. Let's just talk about, for the time being, initiation and to some extent elongation. The DNA polymerases in eukaryotes are Pol alpha plus the primase. Pol alpha seems to serve a similar kind of role as DNA polymerase 1, dealing with Okazaki fragments. There's one big difference, however, with DNA polymerase alpha versus DNA polymerase 1. DNA polymerase alpha does' have that 5' to 3' exonuclease activity. And that tells you need something else to deal with your primers. But DNA polymerase, and basically this is the DNA G. DNA G is that primase. Basically, you can think of poly alpha and primase together, but they are two separate molecules. The equivalent of DNA polymerase 3 in E. coli are Pol delta and Pol epsilon. Whether they really do both leading and lagging strands, people argue about this all the time, but there's certainly two polymerases that are involved in making those long pieces. Those are the ones which are associated with pCNA in terms of making these long processes. Again, pCNA is a sliding clamp. RFC is the clamp loader. This is where we get to our big differences. Fen1 is now what's called a flap endonuclease. And the flap endonuclease, the flap is what happens instead of having something like DNA polymerase 1, which is an exonuclease that chews up your RNA primer. What happens in the case of eukaryotic replication is you end up displacing the RNA strand from the DNA strand. So your polymerase, in this case it's usually DNA polymerase delta, which is extending your Okazaki fragment, will displace the RNA, and you'll end up with this flap of RNA, which is hanging out relative to the usually mostly lagging strand, but of course the primer you have on your leading strand as well. The flap endonuclease clips that off. And so that's one of the big differences between the two. DNA ligase is basically exactly the same thing as we're talking about here. So let's look at the proteins that are present at the replication fork. Here's the SV40 replication fork. The helicase here is labeled as a large T antigen. That, of course, only happens in the viruses. But you've got your leading strand, pCNA, clamp loader, your replicative polymerase, lots of single-stranded binding protein right here. This is your leading strand, your lagging strand. You're going to have a primase together with Pol alpha, which make your primer, and then Pol delta in this case, which is extending along this piece until you get to one of these primers, where you now need a flap endonuclease. And you also have RNase H here, which will take care of it. And again, it seems to be a case of this redundancy, where you've got two different molecules that are taking care of the same problem. This is the way it's drawn in the textbook. It turns out we know that the leading strand and the lagging strand are actually made together at the same time by the same complex. So a better image of this replication fork is actually one like this, because we know that both the leading strand polymerase, which is down here at the bottom, versus the lagging strand polymerase, which is up here at the top, are both together. And there's a really nice animation online, and we're not going to have time to look at it today, but I'll link to it, where they actually show where you've got these leading and lagging strand polymerases, and what happens is this lagging strand sort of trombones in and out of this complex, whereas all the rest of this actually stays together and replicates through the rest of your genome. So that's replication fork. What about origins? Well, you got big genomes in most eukaryotes, and it turns out that the DNA polymerases in eukaryotes are actually a lot slower than the DNA polymerases that you have in bacteria. And that means that you have to have multiple origins of replication. Otherwise, there's no way you could replicate your whole genome. And above and beyond that, most eukaryotic cells go through a cell cycle, where they've got the S phase, where they do all their replication, and then a bunch of other phases, which are important for cell biology. So not enough time. Your polymerases are slow. You got big genomes. Got to have multiple origins of replication. Without multiple origins of replication, you can basically forget it. So how do we know about some of these multiple origins of replication? Most of the work has been done in Saccharomyces cerevisiae, where people found these so-called autonomous replication sequences. Those are shown here. It's a genetic experiment. Basically, you take a piece of DNA and you put a genetic marker on it, and that genetic marker basically says these cells are only going to be able to survive if they can replicate this piece of DNA that has one particular marker on it. And they can only do that if they have an ARS sequence, an autonomous replication sequence, or an origin of replication that you put onto one particular piece of DNA. So all of these cells which have this piece of DNA with this ARS on them grow nicely. The ones over here grow really poorly. They do grow a little bit, but that's a genetic artifact. So these guys say, if you can now get these pieces of DNA out, you can find that they have these autonomous replication sequences that are associated with. Same thing is true, again, for bacterial, eukaryote, archaea as well, though we know a lot less about it. Have to have all of these initiation proteins associated with origins of replication. I'm saying origins because, of course, there are lots of them. First one is the origin recognition complex, also known as ORC, way better for the Lord of the Rings, but no, it's the origin replication complex. Then you also have these two proteins, CDC6 and CDT1, which are basically involved in getting the helicase to the right place on your origin so you can actually start. That helicase is MCM, MCM 2 through 7. Briefly mention where these names come from. Again, origin recognition complex. CDC6 is a classic example of what's called a cell division cycle protein or a cell division cycle mutant. So if you make a mutation in CDC6, you have a problem in the cell cycle. Well, that particular problem in the cell cycle is not being able to replicate properly. CDT1 is again one of these things that was found through yeast genetics. And MCM are the proteins that you need for mini chromosome maintenance. Well, we talked about the ARS sequences. These are the autonomous replication sequences. Well, those are mini chromosomes. If you have a loss of any one of these proteins, you're not going to have these mini chromosomes being maintained. It turns out that these guys have helicase activity. So let's look at one of these origins of replication. Right here, first thing that happens, the origin recognition complex binds to this origin of replication. You have interaction of CDC6 and CDT1. That leads to the loading of the MCM helicase. Once this MCM helicase is loaded, it can now pull apart those two strands of DNA, allows the DNA primase and everything. You get your replication forks, and everything moves along happily. Now, this is true when you want to start your replication. You want to start at the beginning of S phase, but you only want to do it once. And so how do you do that licensing thing? Again, we mentioned in E. coli, if you've got hemimethylated DNA, you know this has just been replicated. How do you know that in eukaryotic cells? Well, here, turns out that the ORC is really the most important aspect of this. So that origin recognition complex, if it's not phosphorylated, then the cell knows this is a place where you want to start your replication. If it is phosphorylated, then it says, hey, I just used this origin of replication. Don't start here. Find somewhere else to go. Or wait until this phosphate has been removed. And the cell cycle, again, hopefully review for all of you, is almost all driven by phosphorylation by the cyclin-dependent kinases. So those kinases, what do? They put phosphates on things. So let's look at this process. Here, again, we've got association of CDC6 and CDT1 with the origin recognition complex. Those then recruit the helicase. But it's that, CDC6 and CDT which are recruiting the helicase. We need to get rid of CDC6 and CDT in order not to get more of these things being associated here. The way you do that, phosphorylation, again by cyclin-dependent kinases. Phosphorylation of CDC6 leads to its degradation through the proteasome that we talked about before. In fact, first thing that happens, it gets polyubiquitinated. That polyubiquitin leads it to its degradation process. And CDC comes off. You phosphorylate here. And this is the process that gives you licensing. Says origin use once and only once per cell cycle. So this is, next slide here is basically a review of what I talked about. I'm not going to go through the whole process here because we're already at 10:05.